the GDL, which is a cyclic ester, it slowly hydrolyzes, forming the parent gluconic acid, this. The acid dissociates, of course, and pH consequently drops slowly. The protons that come from the acid react with calcium carbonate, releasing calcium. But since the calcium carbonate is homogeneously distributed inside the bead, the calcium will be released homogeneously at the same time at all places. So this is a reaction. Carbonate, calcium carbonate plus protons give free calcium and bicarbonate, and actually also some CO2 if the pH goes down too much. So calcium carbonate reacts with the alginate, leading to a slow and homogeneous gelation. Okay, so I will go back a little bit to alginate beads and capsules and discuss a little bit a special application, namely the entrapment of live cells, namely Langerhans islets for treating diabetes. I assume you know the background for diabetes. The Langerhans islets produce insulin when you need it. And people who have diabetes have a badly controlled release of insulin, so something else is needed. And a very famous research paper came in 1980 by Lim and Son. They published in Science in November 1980 that microencapsulated islets, meaning Langerhans islets, could be used as bioartificial endocrine pancreas. So having small gel bead containing Langerhans islets, you could make artificial pancreas. And this is from the abstract. It said a single implantation of microencapsulated islets into rats with streptozotocin-induced diabetes corrected the diabetic state for 2 to 3 weeks. The microencapsulated islets remained morphologically and functionally intact throughout long-term culture studies lasting over 15 weeks. So, I think the whole diabetic world were excited about this. So what happened? There were some problems. Many groups failed to reproduce the results of Lim and Son. Often the beads, when implanted into animal, experienced fibroblast overgrowth because it was recognized as something foreign, and also sometimes inflammations occurred. It turned out that commercial alginate, for instance from Sigma or from some other major supplier, were contaminated with cytotoxic components. And therefore, in the mid-1980s, some groups started to, or some companies started to produce ultra-pure alginate. The other problem is that the beads had a polylysine coating, and polylysine is cytotoxic too. So one solution was to use ultra-pure alginates. But in that work, a surprising result came up in our research group. in the 1980s and 90s. It turned out that some alginates themselves, even if they were pure, could stimulate the immune system. And then, since we had the knowledge about different alginates, we started to test which type of alginate will stimulate the immune system. Does it depend on G content or not? I forgot to mention this, that the beads should contain, of course, non-stimulating alginate coating. So we were looking for what type of alginate is immune stimulating, and are some alginates not stimulating, so we can use as a coating. So another research group in Trondheim led by Professor Espevik, which is the most cited Norwegian scientist in natural sciences at least. He had an essay for studying cytokine production from human blood cells, from monocytes. And he found that alginates are able to stimulate monocytes to cytokine production, and especially TNF-alpha and some of the interleukins. It was later found that some receptors, TLR2 and TLR4, which means toll-like receptor 2 and 4, otherwise known to recognize bacterial lipopolysaccharide, are involved. The interesting thing was that the potency or efficiency increased with the content of M and also molecular size. So that means that alginate rich in M and low in G stimulated a lot. So clearly they were a problem in such beads. But alginates with a high G content, 60, 70, 80%, had almost no immunostimulatory effect. Meaning that we could coat the alginate capsules with another layer of alginate outside the PLL. Another finding was that making short blocks with low activity, short M blocks, by binding them to particles, enhanced their TNF-inducing potency by 2 to 4 log units. So 100 to 10,000 times more effective, which is related to the clustering of alginate on the particle. And some experiment with mice were done, and when given prophylactically and also interperitoneally, alginate with about 90% M obtained from bacteria has been shown to protect mice from lethal E. coli infection and also X-ray irradiation, and to stimulate murine hematopoiesis. So it seemed by the end of the 90s and early 2000s that much of the problems could be solved. So new capsules could be made. And in the mid-1980s, we in Trondheim were involved in a new series of human trials carried out in the United States. So in 1985, a single person who was heavily diabetic got injected directly into his abdomen alginate beads containing human islets.